Virus is a submicroscopic infectious agent that replicates only inside the living cells of an organism. Viruses can infect all types of life forms from plants, animals to the microorganisms, including bacteria and archaea. Virus is firstly discovered as infectious agent by Dmitry Ivanovsky in 1892. Tobacco mosaic virus was firstly discovered by Martinus Benjaring in 1898. The coronavirus is a large group of related RNA viruses which cause diseases in mammals and birds. These viruses cause respiratory tract infections. It belongs to family of Coronaviridae. The virus was isolated from nasal swab and throat swab samples from the suspected COVID-19 patients. Oropharyngeal samples were diluted with viral transfer medium containing nasopharyngeal swabs and antibiotics at ratio 1:4. It is then incubated for 1 hour at 4 degrees Celsius. Coronavirus enveloped with positive sense single-stranded RNA genome and nucleocapsid of helical symmetry. In the structure of COVID-19, particles are spherical and have proteins called as the spike proteins. These spikes latch onto human cells and then undergo a structural change that allows the viral membrane to fuse with the cell membrane. Coronavirus enter inside our body through ACE receptors, that is angiotensin converting enzyme 2 receptors. Present on our lungs, they multiply inside our body using our own cells.
COVID-19 can be diagnosed by two tests. First, viral test, which include RT-PCR. The second test is serological test, which include antibody test. Viral test include RT-PCR, which is most widely used. PCR, that is polymerase chain reaction, is invented by Carey Mullis in 1983. This test is used for the amplification of a DNA sequence based on the principle of enzymatic replication of a nucleic acid. But as COVID-19 contains RNA as a genetic material, RT-PCR technique is used. RT-PCR, that is reverse transcription polymerase chain reaction, is one of the most accurate technique for detecting and studying coronavirus. It includes transcription of RNA into DNA by reverse transcriptase enzyme. The sample is collected from the parts of body where coronavirus gathers, that is nose, throat. This sample is then treated with various chemical solution to remove proteins and fats, as a result of which only RNA remains in the mixture. This RNA is then transcribed into DNA by using specific enzyme. Then scientists add some short fragment of a DNA which is complementary to viral DNA. Then this mixture is kept in RT-PCR machine to make multiple copies of DNA. This is indicated by fluorescent dye. The presence of DNA indicates that the person is suffering from COVID-19. The second test used for the COVID-19 diagnosis is serological test, which include antibody test. It is carried out when you had previous infection. For this, blood sample is collected to detect the presence of antibody. Antibody is the protein which is produced by our immune system to fight against pathogen. It takes 1 to 2 weeks to produce antibody in our body after you become sick. The presence of antibody in our blood indicates that the person is suffering from COVID-19.
The new coronavirus is a RNA virus. It is a collection of a genetic material packed inside a protein shell. As it is a virus, every virus mutates, and mutation, it is a part of virus life cycle. And mutation means alteration in the nucleotide sequence of the genome. Once RNA makes contact with the host, it starts to make new copies of itself, and that can go on to infect the other cell. As already we know that the coronavirus seemed to be mutating at a high speed, and it was reported that the virulent Yale strain of the coronavirus is responsible for high mortality. Though no evidence of the same has been found as it. It was also found that the A2A type of mutation, which is become more predominant also increase the efficiency of the entry of the viruses into the human body through the cell. Due to this type of mutation, there is a difficulty to find out the vaccine against the COVID-19. For vaccination against COVID-19, researchers are focused on analysis of mutation. The researchers are currently working on vaccine targeting the strain severe acute respiratory syndrome.
Hydroxychloroquine. It is an allopathic drug. Mainly, it is used in the treatment of malaria-like diseases. WHO has listed hydroxychloroquine as an effective drug against COVID-19. Plasma therapy. In search for an effective treatment for COVID-19, an old method is resurfaced, that is plasma therapy. Basically, plasma is a component of a blood. Mainly, plasma contains an antibodies which can recognize and fight against the pathogens and such viruses. The blood of a people who have recovered from an infection contains such antibodies. Now, I will explain what is a plasma therapy. Doctors take out the blood from a recovered patient and separate the plasma and insert it into a people who are currently fighting with the infectious diseases like a COVID-19. This can help their immune system to fight with the virus more efficiently. So recently, researchers and healthcare are looking into the possibility of using this method to treat the COVID-19 patients. On 5th of June, plasma therapy has successfully done in Kolhapur, Maharashtra.
The contribution of microbiologists in this pandemic situation is very appreciable because they guide for how to treat the patient by their research. They suggest by their study to use the efficient or more useful medicine against COVID-19. The most important work that they are doing nowadays, that is vaccine production against COVID-19. In the case of COVID-19, there are many unknowns that microbiologists are all working hard to discover. They are at the forefront of this type of infectious disease outbreaks. They conduct critical research to identify the infectious agent, study their reproduction and biology, develop and test vaccines and antimicrobial drugs, making them available to the broader medical community for treatment and prevention.